Okay. Buenos días a todos. How are you doing, guys? Hanging in there?
I'm good. You?
Good. Good. I started a migraine 2 days ago. I'm still alive, but anyways.
Oh.
It can always get worse.
Yes, it can. I'm trying to do everything to keep it. Like, taking my medicine, which I hate. I dislike the taste. The taste just brings me my hairs up. I'm like, ugh. But it's the only thing that works. I can take Tylenol and whatever. That doesn't make any change. But anyways, I'm gonna start by saying I posted in the lab news because we have seen plagiarism in the lab reports. And I talk about it in the first lecture. It is in the syllabus. This time, you are given 0 for those answers where you plagiarize. Copy-pasting from an article or something, and then given the citation is still plagiarism. Okay, you need to paraphrase. Next time we get it, is a 0 for the lab report. Okay, I put the note, the link to the student, academic behavior, I think is called the policy. So please read it so you are aware of the consequences that this can carry out for you guys. I am not doing anything that is not within those policies. I've been lenient this time and giving 0 to that answer because I have already spoken to you about this. It's in the syllabus, so you should always read the syllabus and go to see the links and all that. Next time is a 0 for the lab report. Okay, this is a 3rd, 4-year level course. You should have gone through this in every single lab, right? And so it's not acceptable. It's a behavior that if we don't correct it, it stick with you guys. And I want to believe that you all are honest people. Okay, that's my pre-assumption always. You are good people. And sometimes, oh well, I didn't pay attention. Okay, now I'm telling you, please be good people. And that starts with such a tiny behavior of not plagiarizing. Okay, I sometimes write things that I have written before, and then oh my God, I need to change it. Even if myself who wrote it first, I'm plagiarizing myself. So I always need to paraphrase. So I always aware of that. So you read on a paper, you need to paraphrase when you put it into the paper. Okay, so just a little gestures like that make us a better people. Just be more conscientious about it. Okay. Okay, so we are going to need to go to my fish stuff. So today we are getting into the fish. Development. There we are. Now I have it on the other screen. Okay, so here we're going to see a little bit of variation in the way how fish develop. And recall that amphibian embryos divide by a radial holoblastic cleavage, right? So the whole thing, the whole egg is divided from the animal pole to the vegetal hemisphere, okay? And it goes into perpendicular and Just wait a second to allow somebody to come in. And then we have meridional and equatorial divisions, right? That's exactly how we were having first meridional, second meridional, and then equatorial displays. Now, in fish, birds, reptile embryos, we have a cleavage that is meroblastic, okay? Which means that only a small portion of the egg cytoplasm will be used to make the cells of the embryo. In mammals, we have a holoblastic cleavage, which is modified to make the placenta, which will enable the embryo to develop inside the mother. But for fish, bird, and reptiles, it's meroblastic. Only one section of the embryo, of the egg, will be divided. So in this first image, we have the first steps of development and cleavage. And so as you saw, we talked last time in the lab, zebrafish, Daniel Verrio, is a really good model organism to study development for vertebrates because of several reasons that I think you needed to write in your lab report for tomorrow, Saturday, that you need to submit. So they have large amounts of egg production. They breed year-round with the right conditions if they are well fed, and all that. Of course, this is under laboratory conditions. They have transparent eggs. So this is something that we can see in this one, but they have transparent eggs. So we can see the embryo development as it's happening. And they go from one cell, like the one we have in here, to a larvae that we have in here in about 24 hours. So here is a 24-hour post-fertilization, and we have an embryo, a larvae, okay? So it's very, very, very quick. And normally 48 hours to 60 hours, more or less, they hatch. Okay, so it's very, very quick that happens. So we go from one cell to two, and so on. And we can see exactly the movements of the cells. And it's so beautiful just to see it. As I said, this is something that I would have been able to see if we had a lab in person this year, because I always wear them for this lab. So we can easily follow the cells as they continue to move, go through all the different movements in the egg. So in here, fish eggs are, say, telolithal, which means that most of the egg cell is yolk. Okay, we have already defined this a couple of weeks ago. So cleavage, if most of the egg is yolk, cleavage can only occur in a little region that we're gonna call the blastodisc. This is what we see here on the top. Okay, so the blastodisc is a thin region of yolk-free cytoplasm and is always placed at the animal pole. Okay, so in here we have, this is the stage 0. This is where the egg meets the sperm. So we're gonna have some sperm, and we know that the sperm, like we just wanna go to, Jesus, we went really fast. I just wanted to do this. So you see this little thing here, which says MP, that's the micropili. That is the region where the sperm got inside of the egg. That's the tunnel that the sperm made in order to get into the vitelline layer, okay? So when the egg is stimulated by the entrance of the spermatozoa arriving and entering through the micropili, we're gonna have the first reaction to polyspermia, which is, what is the first stage to avoid polyspermia? Once the sperm enters into the egg, what it was? And I see your mouth just going, I know. Remember the calcium reaction? Okay. So that's the first step. So the sperm gets in, we have the calcium reaction that is caused by the release of the cortical granules, right? That we have in here. And we have here the progression of the calcium. So the white section here is how that wave of calcium is going from the animal pole to the vegetal pole in here, okay? So at this point, they're gonna cause a lot of things. One is the exocytosis of those cortical alveoli that we have seen before to avoid polyspermia. That's the initial reaction, okay? And with that, what happened, remember the mucopolysaccharides cause an osmotic reaction that brings water in and creates a fertilization membrane, right? And this is the cell membrane. Now, all these reactions also cause a stimulation of the actin cytoskeleton that's gonna cause an squeezing in of the yolk, okay? So the section that has no yolk gonna start moving up into the animal pole, okay? And this is because of the cytoskeleton stimulation due to this exocytosis of the cortical granules. So this is going to cause a change in the shape of the egg. Initially, it was round, but after the cortical reaction and the calcium wave, all the non-vitelline part of the vitellus goes on the top, and it becomes more like a pear. You see here the little pear shape? Okay? So all that is caused just because of the entrance of the sperm. Okay? So in here, once the wave reaches the vegetal hemisphere, we get into stage 1. Okay? You see here, the yolk droplets are very well distributed all around. And in here, again, I'm doing the same thing. In here, you see that little piece that doesn't have yolk, right? That is what it will become the blastodisc. So it takes a little bit of time to get from here to this little mole on top of the animal pole. So in stage 2A, in here, the male and the female pronuclei start migrating to one another and gonna fuse together, okay? At the center of this cytoplasmatic disc that form because of this wave. And we see here that it continues to accumulate some cytoplasma that doesn't have the yolk droplets, okay? Chromosomes start dividing into 2 groups at the poles of the spindle, marking the end of that stage. And all of this happened during this stage 2A. So fusion of the pronuclei, placement of the spindle, and then we start going to each side, okay? We also see another change that occurs during this stage 2A, and that is these yolk droplets, okay? So this is these droplets here. Look what is happening. They are migrating towards the vegetal pole, okay? So we have some oscillation of contractions that continues to occur in here to make these droplets to move here to the vegetal pole, while more of this cytoplasma continues to accumulate into the animal pole, and then it becomes a little bit of convex shape. At this stage, this is becoming the blastodisc. Here is the apical blastodisc, but here it becomes really the blastodisc, okay? Now, in stage 2B, the layer of cortical cytoplasm covering the yolk sphere is very thin except where it forms the cat-shaped blastodisc. This piece here is a little thicker, okay? And this one here is really, really thin. And at the end of this stage, all these droplets from the yolk start migrating really towards the bottom, okay? So by the end of this stage, stage 2B, most of the droplets from the animal hemisphere have already migrated to the vegetal, and 2 dimples will appear in our blastodisc as marker of where the future blastomeres will be placed. So cleavage now gonna take place. Now we have fused the 2 pronuclei, all the droplets gonna go down, and we're gonna have another sections that are important here. So the cell divisions do not completely divide the egg, right? It's only in the little mud that they have. This is the pear shape that I was mentioning, okay? And this is then the blastodisc, right? And cleavage only occurs at the level of the blastodisc. And because of this cleavage is said meroblastic, only occurs at the level of that blastodisc. So this blastodisc is what will create the embryo. And because the embryo generates from it, it's also said to be discoidal because it only comes from the blastodisc. So fish cleavage is called meroblastic discoidal or discoidal meroblastic. Okay. So as we see in B here, the first cleavage is meridional, right? So perpendicular to the axis. In C is our second division, okay? And this also meridional. Same thing as we have seen in the front, perpendicular to one another, right? 1 and 2. Then, so we have 1 cell, 2 cell, 4 cell. In D, now we have a second division, a third division, and that is an 8-cell embryo. So we have 2 rows of 4 cells. So we have here only 4, and then now we have two rows of 4 cells. So in E, we have a 32 embryo. And this one was here, we see when we start the equatorial divisions a little bit more, okay? And then in F is a 64 cell embryo, okay? And then something that happened from this division on is that we lose a little bit of the synchronous divisions. And then we're gonna start having different axis angles at which these divisions are occurring. But as of the 64 embryo stage, we don't call this anymore a blastodisc, it becomes the blastoderm. So now I call it blastoderm, it becomes a blastula because we have now enough number of cells in here. Another difference that we want to see is the cell movements. In the mid-blastula transition, it will occur normally about division number 10, okay? So we have about 500 of cells, okay? The 10th division is the point at which gene transcription starts. Not before, it's about mid-blastula, 10th division. So we're gonna have the transcription of genes starts there, and then cell division is slowed down. At the beginning, it's really, really fast, and then once transcription starts, then cell division gets reduced. And because now we have transcription factors that will determine the fate of most of these cells, We're also going to start seeing some cell movements, okay? So in this figure, I'm going to just define a couple of terms first. So we have 3 different cell types in the fish blastula. The first one is the yolk syncytial nuclei or layer, okay? So it's all this section here, the yolk syncytial layer, which contain nuclei, okay? Normally, I'm going to refer this as the YSL, yolk syncytial layer. This is the fusion of cells at the vegetal edge of the blastoderm. So this is the blastoderm, right? And these cells are placed just underneath, just beneath the blastoderm, okay? And they produce a ring, a little ring around. So these are here the cells, and they're all around the blastoderm, okay? Some of these nuclei will eventually move under the blastoderm and will become the internal yolk syncytial layer. Don't worry, we're gonna see them in a couple of slides, okay? While some will become the external yolk syncytial layer. So some gonna go inside here and some gonna just remain on the outside of this blastoderm. The second group of cells are the enveloping layer. It's made up of the most superficial blastoderm cells, which form the epithelial layer, and it's just one cell layer, as we see here in the green. So it's just one layer, one cell layer, all here in green, all that edge. The third group are the deep cells, and deep cell layer is between the enveloping layer and the yolk syncytial layer, and this is what will give rise to the embryo. So 3 important layers: enveloping, deep, and yolk syncytial, and the deep cells are the ones that are going to generate the embryo. So this image allows us to see that at the blastula stage, most of the fate of this blastoderm are not necessarily yet determined, okay? Their fate appears to be fixed shortly before gastrulation will begins. And this is gastrulation. So we just got these cells placed. This is the ring of the yolk syncytial layer with the nuclei. This is the deep cells, and this is the enveloping layer. Okay, so we just have all these things in place. So in here we see that the fate map of the deep cells after the cell mixing has stopped. We have already a little bit of an idea of where the different organs are going to be forming, okay? So ectoderm, mesoderm, endoderm, and this is just the yolk that will nourish that embryo while it develops, okay? So it's not all of this, it's just this section over here. And at the same time, we have already determined which one is the dorsal side of the embryo and which is the ventral side of the embryo. Okay, so in the next image, we want to see gastrulation. So during gastrulation, the first movement to appear is epibole. So remember, epibole is the covering of the most external cells, and they're covering the egg. Okay, so it's like putting some tight socks on your feet, right? So it starts from the top and goes all the way around in this case. Okay, so cells of the blastoderm, that's the section here, will cover over the yolk. So epibole is the first movement. As the cells start going all around, we have here a section on the yolk syncytial layer, the interface between the yolk and the blastoderm, that's going to start to squeezing in. And you see here how we change a little bit of the shape of the egg, right? So, and this is what we're going to call, it's like a little belt, okay? And this will be the germ ring. In the lab, we always are able to see this so nicely, and we see how this dome here, it's a stage that is called the dome in the zebrafish development, start going inside. And this is squeezing in caused by the epibole, gonna cause a mix here. And we see how, remember that I said that some of the nuclei of the yolk syncytial go inside and become the internal yolk syncytial layer? This is how it goes in, because by the epibole we're squeezing in these cells, okay? So at one point during this movement of gastrulation, and normally it gets to the what we call the half epibole stage, 50% of the epibole. So when the 50% of the egg is covered by the epibole movement of this enveloping layer are going down, then at one point we are going to have a thickening side of cells, okay? And normally it occurs in the dorsal side of the embryo. So this is the dorsal and this is the ventral. This is the animal and this is the vegetal pole. So one side of the blastoderm becomes thicker. This is what we see in here. And this thicker side becomes the dorsal side. And that dorsal side, it will be the equivalent, as I'm gonna say that later, of the dorsal blastopore lip. Remember in the frog, that's the organizer, okay? So this is what we have here in this germ ring, because this is all around that. And here we have other type of movements that we're gonna see in a couple of slides, okay? So after the blastoderm cells have covered half of the egg, so the 50% blastula, okay? We're gonna see some cells that start epibolizing that blastoderm at the margin. So we're gonna have some cells that start continue going. And here we have a little bit of zoom in of this section. And we're gonna have cells that are part of the deep cell layer start ingressing. Remember that movement, ingression? Okay, so they're gonna duplicate the layer. So we have one and here we have two, okay? We're also gonna have some cells that gonna start involuting. Involuting was the rolling in, right? So in here they're gonna, because these layers are dividing, they're gonna roll inside. And these cells at this edge of the germ ring that are involuting will become the hypoblast that will structure the mesoderm of the embryo, okay? So ingression to increase the number of cells that are gonna form that embryo. Involution, they're gonna create the mesoderm as the cells roll in, and all of this is from the dorsal side. So dorsal side, dorsal lip of the blastopore, and this is gonna become the shield, okay? We're gonna get in there. We also have a group of cells that are not involuting, they just stay there, okay? So these cells that are involuting will become the hypoblast. All of these, up to here, start occurring within the first 6 hours after fertilization. So it's very quick, okay? Okay, now we are 9 hours. We see that epibole has continued, right? We see that epiblast, and we have here the hypoblast. Okay, the epiblast will be the presumptive ectoderm, the hypoblast will be the mesoderm, and so we're in here, we're going to have the endoderm. So as the ectodermal epiboly nears completion, this is about 75-80% epiboly. Okay, the hypoblast start projecting towards the ventral side. So it continues to roll in over, it continues to go inside to become that mesoderm. And of course, it also has the endoderm precursors, okay? And they start covering the yolk, as we see here. Okay, so it continues to go in this direction. Here in D is 10 hours later of post-fertilization. And at 10 hours later, we already have the 3 germ layers: ectoderm, mesoderm, and endoderm. Here we finish up the epiboly of the cell. So this is a 95% of epiboly. Okay. All the hypoblast mesoderm went up. And then we have, at this point, this is the trunk, this is the head region, and this is the ventral portion, ending up here with the tail bud. Okay. So we have all the things placed now where they should be 10 hours after fertilization. Now, we have some movements that are occurring, as I mentioned a couple of minutes ago. So once that hypoblast has been formed, and I mean hypoblast is this, right? The one that has the mesoderm. So once that hypoblast had formed, cells of the epiblast and the hypoblast start intercalating, and those are the movements that we see right here, okay? So they start intercalating on the future dorsal side of the embryo, and that is going to cause a thickening in that dorsal side, and this is what we call the embryonic shield, okay? So epiboly, we say that it started going from the animal pole to the vegetal pole over the yolk. We had involution. We have the involution, the cells going inside, duplicated, or ingression, right? Ingression and involution. Both of them are happening. So here we have involution only, okay? And that will generate the hypoblast. Then we have another movement that is convergence at that thickening section. A convergence alongside with extension. So this starts squeezing in from the sides, from the lateral sides, going to the dorsal, and then extending towards the animal pole, okay? And that brings the hypoblast and the epiblast cells to the dorsal side of the embryonic shield, because they were all around, so they're gonna go come and squeeze in. So here is the shield, this is epiboly, this here is involution and ingression, right? And in here we have the convergence, because we start bringing in the cells, and we have the extension, okay? This embryonic shield is the functional equivalent to the dorsal lip of blastopore that we saw in amphibians, okay? So hypoblast cells converge and extend anteriorly, and eventually will become something that we call the chorda mesoderm. This chorda mesoderm is the precursor of the notochord. So highlight that term, chorda mesoderm, because we're gonna see it again and again in the future. Chorda mesoderm is the precursor of the notochord. We're gonna see it in the lab many times, so I just wanted you to sync it now, okay? Now, the cells that are adjacent to the chorda mesoderm, so just on the sides of this, will become the paraxial mesoderm. The paraxial mesoderm is a transient structure that will become the somites. And the somites will become then the vertebra and all of that. Okay, so keep that in mind. Anything that is on this each side of the chordal mesoderm is the paraxial mesoderm, and they will become the somites. Now, the epiblast cells that cover and extend will become the neural keel at the dorsal midline. Okay, cells remaining in the epiblast become the ectoderm, and endoderm normally arises from the marginal cells of the late blastula. The axis formation, it occurs in a very similar way as we saw in amphibians, where in this case, this is the embryonic shield, and this section, right, is critical in establishing this dorsal-ventral axis, exactly the same thing as we saw in amphibians. Okay, so this embryonic shield forms the precaudal plate and the notochord. So some researchers made very similar experiments as the one that we saw by Spearman and Mangold with the newt, remember? So they take an animal donor, they take that embryonic shield from this one and transfer it into the ventral portion of a host and see what happens, okay? So what we have in here is that we have an embryo that was forming two axes, okay? So the embryonic shield acquires its organizing ability in the same way as the amphibian organizer. Fish specify the epidermis by using the bone morphogenic proteins, BMPs, in the same manner as we saw in amphibians, while WNT proteins induce the ectoderm, and this is what we're going to be seeing in the next slide. So the signals secreted by the zebrafish shield are the same as in Xenopus. TGF-beta superfamily members, which are nodal and BMP. Okay, so both of them function to induce differential fates of the germ layers across that dorsal-ventral axis. So we saw a couple of molecules when we talk about the frogs. Particularly, we talk about cording, right? But we also talk about noggin, that we want to see in the next slide, and follistatin. So those three signals are secreted by the shield, exactly in the same way that they were secreted by the dorsal lid blastopore, okay? And all of them serve to block the ventral induction to promote the neural development. So in here, we have nodal and we have BMP. Okay, so BMP and nodal signaling determine by different interactions which of the axes are going to form. Okay, so nodal normally promotes the endoderm and mesoderm. Okay, we have seen that before. And then we have the BMP, induces the ventral fates of all germ layers. So, and normally as a function inhibited by cording, and we see it here, cording causing that inhibition. Okay, so all the ventral fates are of the competence of BMP. Okay, and here we see how nodal or VMP receptor normally are controlling the downstream action of phosphorylated SMAD, and then SMAD are the ones that are going to trigger the transcription factors to determine the mesodermal and ventral genes. Okay, so here we have our egg. This is just the pattern of expression. in zebrafish. This is the animal, this is the vegetal, this is the ventral, and this is the dorsal. Nodal, look at where it is placed, just right in the middle, in the interface between the animal and the vegetal. Then we have BMP, always expressed in the ventral portion. So this is what they determine the ventral phase. And then we have chordin. And chordin, excuse me, is placed right in the dorsal section of the embryo where the shield is determined. Is this a sketch clear? Okay. So there's a couple of experiments that have been made just to corroborate that nodal and BMP, both of them are really important in the formation of this dorsal-ventral axis. So here what we have, SH represents the shield. So that dorsal lip, that organizer. This is the dorsal side, and this is the ventral side. We see that nearby the shield, we have a higher expression of nodal, while in the ventral portion of the embryo, we have a high expression of BMP. So, as we see in this figure, BMP higher in the ventral, while nodal higher in the dorsal side. What we see here in B is the correlation between the relative proportion of BMP and nodal secreting clones, okay? So when nodal and BMP are normally in the same vector, this is the vector nodal and BMP, then the dorsal-ventral axis of the embryo, we have the expression is in the same direction, right? So we're going to have the first embryo is this one, and the second is this one. So both of them are going in the same direction, are parallel. Here in C and D, what we have is when the nodal BMP, as we see it in here, okay, is perpendicular to the original dorsal-ventral axis. We see that it's perpendicular, okay? The secondary embryonic axis grows perpendicular to the primary axis. So it goes this way. In E and F, what we see is that we oppose, we switch when the nodal BMP vector is against of the original dorsal-ventral. So dorsal-ventral goes to the left and nodal BMP goes to the right. The secondary axis grows in opposite direction. So here is where this embryo is forming this way. So we know that nodal is always placed in higher concentration nearby the shield, and BMP is in the opposite direction towards the ventral portion. Well, fish has another center that is called the mucous center. We also talk about this mucous center in amphibians, remember? So the head organizer, the shield, we have here that while the blastopore lip cells are 180 degrees away from the shield, the tail becomes a little bit also of the organizer. So we have here the integration of BMP and chordin. Chordin, we have two genes that are going to relate for chordin. Appears to be both coordinated by a molecule that we have seen again and again. Remember how many times I told you that we're going to be talking about this, then beta-catenin as being one of the most important molecules in every organism. Here we have again beta-catenin. In this case, activates all of these nodal genes. Okay, so in addition, beta-catenin also activates genes that encode for fibroblast growth factors and represses BMP and WNT expression in the dorsal side of the embryo, because BMP needs to be in the ventral side of the embryo, so we can activate. And what they're going to be activated is the same that we have seen in frogs, right? Noggin, gosicoid, and decuff. Okay. In zebrafish, remember those nuclei of the yolk syncytial layer that I mentioned before? So we have here that the nuclei of the yolk syncytial layer beneath the cells will become the embryonic shell. They are the ones that accumulate this beta-catenin, and that is why it's important for this nuclei to be in the ring and then go inside of the embryo as gastrulation movement are made, because this accumulation of beta-catenin are essential for the expression of the nodal genes. So in this way, the presence of beta-catenin will distinguish the dorsal yolk syncytial layer from the lateral and the ventral regions. Beta-catenin then, as we see here, activates Quin, Bususuk, okay? And these two proteins activates the organizer-specific genes. On the other hand, the STAT3 gene, normally the product of this is what favors the cell movement that occurs during gastrulation. So beta-catenin, once again, if we generate a knockout that impedes those nuclei to accumulate beta-catenin, we won't be able to form an embryo, because it activates the genes that are absolutely necessary for gastrulation and the determination of the dorsal-ventral side. So remember, again, noggin induces the dorsal ectoderm to form the neural tissue. And on the other side, cordin, as we see here, two that control for this, activates beta-catenin at the same time. That's a positive feedback that occurs and prevents their complexing with the receptors. So very important beta-catenin. Okay, so that was for the dorsal-ventral. Now we have the right and left axis. So we know, and I said it before, despite the symmetry of most vertebrates, the internal organization of organs is not symmetrical. The heart in most vertebrates is a little towards the left, but other organs are placed more towards the right, okay? So the left side is signaled by the expression of nodal-related genes, and we have seen that before. Here, we have in the middle something that we call the nodal cilia, okay? This nodal cilia is placed in the Kupfer's vesicle, okay? And this Kupfer vesicle, and we're gonna see later the Kupfer vesicle in the lab, okay? You need to identify that Kupfer vesicle. Creates a current that releases calcium, and this calcium stimulates the Notch and BMP pathways, and these two activates PTX2. Okay, PTX2 is a transcription factor only on the left side of the embryo, and it's located in the mesoderm. On the other hand, FGF, the fibroblast growth factor, is expressed only in the right side of the embryo. So you're gonna see, when we're gonna be looking at the heart formation, we're gonna see how this transcription factor, PTX2, is really important for the buckling of the heart and making some organs going into the left side of the embryo. So that's why our heart is more placed towards, a little bit towards the left, because of this transcription factor, okay? Okay, now this anterior-posterior axis. Nothing is simple during development, and the establishment of this anterior-posterior axis is highly cooperative, and we see here all of this cooperation happening. In zebrafish, this is the matter for this anterior-posterior is on the incumbence of FGF, WNT, and retinoid axis in a very similar way as we saw in Xenopus, in the frogs, okay? So first of all, WNT signal represses the expression of the anterior genes, okay? So this is what we see here in blue. These genes are repressed, okay? And here we see WNT, how it exerts a greater degree of inhibition, right, compared to the FGF. So we have WNT will repress the anterior genes that are in the blue section. Retinoic acid, the RA, in here. Retinoic acid is a molecule that is so polyvalent. In this case, it acts more as a morphogen because it's a gradient, okay? So retinoic acid acts as a morphogen, and FGF is required to activate the posterior genes, okay? So retinoic acid and FGF is for the posterior genes. There are a little bit more in red. So here we see how retinoic acid gradient enlarges as the movement, the cell movements are progressing, and particularly the epibole, because the epibole is the one that is covering the yolk. You see how that retinoic acid just goes stretching, the gradient goes just wider, okay? So in this figure, we see an element that is the CYP26 here and here, okay? It's a gene that encodes retinoic acid for hydrolysis. It's an enzyme that degrades the retinoic acid. So in A, this is the blastula stage. This is the late blastula stage. We see how this CYP26 gene is confined into the, I've been talking since 8:30, giving lectures on my throat now. I start getting a little dry, sorry. Is restricted a little bit more towards the anterior region. And this is due to the presence of the FGF and the WNTs. Okay, so in the anterior portion, in the late blastula. In B, as gastrula start going and the cells start epibolizing and getting into the 50% epibole here, convergent extension start taking place, okay, at the margin. And this is farther, so this is the FGF and WNTs gets confined right here in the middle, okay? And then retinoic acid start accumulating in this region. This is the belt that we had before, okay? And start activating genes that are responsible for the posterior neural ectoderm by the end of the gastrula. So we have a formation of the anterior portion first. As gastrula movements start occurring, we're gonna have a greater section where retinoic acid is present, and then we're gonna start activating the genes responsible for the posterior neural ectoderm. Is this image clear? Yeah, okay. Okay, so let's see this in a flow chart. So cells of the anterior part receive very little retinoic acid compared to the posterior parts of the embryo. This is very important during brain differentiation because it starts in this anterior-posterior axis. So we're gonna see this when we're gonna have a full lecture just on brain formation, okay? So FGF positively regulate retinoic acid accumulation by inhibiting the expression of this CYP26. Okay, as mentioned before, this only degrades retinoic acid. So this is what we have this here. Okay. So, but at the same time, that FGF and retinoic acid enhancing the synthesis for enzymes that synthesize more retinoic acid from vitamin A. So that's what is really important, to have a very good input of vitamin A. So we have that retinoic acid formation. In the posterior portion of the embryo, FGF and WNTs repress the expression of CYP26, which degrades retinoic acid, okay, but enhances the expression of aldehyde 1A2, which encodes the enzyme for the synthesis of retinoic acid. So this is how it gets inhibited, okay, but enabling retinoic acid to accumulate and activate the posterior genes, which are HOXB1 and MEIS3. So repression of CYP26, but then enhancing the ones that increase the retinoic acid. So if we go back in here, this is what we have in here. We have this expression here that increases the expression of the genes for the posteriorizing section. So despite different cleavage patterns as a result of the different yolk distribution between fish, which is metaplastic cleavage, and a huge yolk cell with the syncytial nuclei, amphibians have holoblastic cleavage and a blastocele. Here we don't have, right? We don't have that blastocele that we saw in amphibians. However, both types of embryo use similar molecular tools to undergo cleavage, gastrulation, and axis formation. In both amphibians and fish, pattern of the neural ectoderm and mesoderm appears to be controlled by WNT proteins. Both uses beta-catenin for the expression of the organizing genes. Both use BMPs and WNTs to lateralize and vegetalize the embryo, meaning the dorsal-ventral and apical vegetable poles. Both also use a particular WNT protein later in development to posteriorize ectoderm from the trunk neural tube. And with that, we are done with the fish. I just need to go now. Just need to switch my presentation. Okay, so now we are going to see the development of birds and mammals. And we put them together simply because they are very similar. They develop in a very similar way. Just give me a second. I'm having a hard time just retrieving my other screen. Okay. So birds. Come on to this. There we go. So the amniotes are vertebrates whose embryos form the amnion, which is a watered sac, simply. So we see this in reptiles, birds, and mammals. As we see in this figure, the amniote egg displays a set of membranes that enable the embryo to survive on land. Okay, so here we have our egg. That's the dissection of the egg that you needed to do. Okay, so we also see here the yolk sac, very big in birds, not so big in mammals. Okay, so this yolk sac contains nutrients that want to be uptaken by the embryo through the circulatory system to nourish the embryo. And this is one of the big differences between the birds and the mammals, is that if we don't establish that placental connection, this little thing here not gonna last 9 months, right? Then we have a second membrane that is the chorion, here and here. The chorion contains the blood vessels that allow gas exchange with the external environment. So that's the function of the chorion. Then we have a third, that is the allantois here, and just tiny here. Okay, the allantois normally is placed at the posterior side of the embryo. Okay, and their function is just to store waste products. And then we have the amnion here and tiny over here. The amnion enables the embryo to float, to feel cozy in the fluid and avoids desiccation. Okay, so all of us have it. The birds is a little smaller compared to the human that is huge. So how this happen? How this cleavage occur in birds? So birds' eggs are said to be telolecithal based on the yolk distribution. So it's huge, right? A huge amount of yolk, like in fish. It's also blastodisc. So when I have a small disc of cytoplasma, always as we saw in fish, placed at the animal pole, just on top, sitting on top of the yolk. So this is our blastodisc that you needed to see in the yolk dissection. Okay, so birds display, again, as we saw in birds, a discoidal meroblastic cleavage, because it only happens in the blastodisc. The yolk don't get divided. So in birds, the first cleavage is what we see in here, appears centrally in the blastodisc. And then the other cleavage follow to create a single layer. So we have here 3. So this is the first, then second, and then fourth, and so on. So from here to here, we get that in the middle, we have a formation of what will become the blastoderm. So blastodisc is at the beginning here, but once it starts segmenting and segmenting, this becomes the blastoderm. Okay. So these cleavages do not extend into the yolk cytoplasm. So the early cleavage cells are continuous with each other and with the yolk at their base. And this is what we see here in F. You see, cells are not completely separated from the yolk underneath. So this is the blastodisc, one single layer of cells, and this is the yolk. Okay. After this, we're going to have equatorial and vertical cleavages that will divide the blastoderm into a tissue that will become about 5 to 6 layers thick. And these cells become linked together by tight junctions, but we're going to see that in the next slide. So in here, I think this image is from the 7th version of Gilbert. The new one, the 12th edition, normally are the ones that are now in the 12th edition. This is the 7th, but I prefer this one. It's bigger and allows me to show a little bit more. Okay, so here we have the blastoderm that generates through the divisions of the blastodisc. Okay. And here we have different things that you gotta really pay attention, because in the lab, I'm gonna be asking you to identify those sections, okay? The yolk is here, and between the blastoderm and the yolk, we have the subgerminal space, okay? This space is created when the blastoderm cells start absorbing water from the albumin that is surrounding and secreted between themselves and the yolk. They require this space to appear here. This space is going to play a very important role because the cells that are found in this blastoderm going to start losing connection, going to start dividing, okay? And going to create the hypoblast cells. So at this stage, the deep cells in the center, so this one to here, going to start being shed, some will die, and then they're going to leave behind a one cell thick area in the middle that is called the area pellucida. Okay, so these cells, so we are here in this step 1. This is the little section in the middle, right? They're going to start duplicating and then become the zona pellucida, right in the middle. This area pellucida forms most of the actual embryo. So this is more equivalent to the deep cells in the fish. Right? So this is the area pellucida, which will form most of the embryo. The peripheral ring, so here and here, so what we have left on the edges, have not shed their deep cells and constitute the area opeca. So we're going to have that section here and that section here. That's the area opeca. Now, between the area pellucida and the area opeca is a thin layer of cells that is called the marginal zone, or the marginal belt. So between this and this little zone over here, okay, the dark gray color in there. Some of the marginal zone cells become very important in determining the fate during early chick development. So blastoderm, the blastoderm middle gonna duplicate, some will die, some will be shed, and then become the area pellucida. These cells also want to absorb water from the albumin to put it into the space between the blastoderm and the yolk to create the subgerminal space. These cells that have no shed from the edges are the area opeca, and we're going to have one little zone that will be the marginal zone, or belt, because it's all around. Now, when the egg is laid, the blastoderm contains about 50,000 cells. So it's a lot of cells, okay? At this time, most of the cells of the area pellucida are still at the surface, forming that epiblast. So that's the epiblast in here, in the area pellucida. Okay. Some of the area pellucida have delaminated. This is what we see in here. They are delaminated and migrating individually into the subgerminal cavity. And these cells normally going to start to polyinvaginate into islands. So little group of cells, between 5 to 20 cells. Normally they're going to start aggregating in this section. Okay. And this cluster is what will become the primary hypoblast. So cells from the epiblast on the zona pellucida will delaminate and create the primary epiblast. Next, we're going to have a sheet of cells from the posterior margin. Remember that belt that we have in their section? So it's recognizable from the other regions of the margin because of the localized thickening. So once again, as we saw in fish, we're going to start having a thickening. And that thickening always occurs in the posterior part of the embryo. This region that thickens is called the coracycle. This group of cells start moving anteriorly and start picking up the island, the primary mesenchyme from the delaminated cells from the epiblast, and it goes to the next, to the opposite end. Once they are consolidated, the cells from the coracycle with the primary mesenchyme here, this becomes the secondary hypoblast. So now we have a two-layer blastoderm, right? Epiblast and hypoblast. Okay. These two are joined in the area opeca. Remember, this is the area opeca here and here. Okay. And although the shape and formation of the avian blastoderm differs from what we saw in the sea urchin and amphibian, the spatial relationship is more or less the same. Okay. So here we have that all three layers of the embryo are proper established. So a lot of the extraembryonic membranes are formed from cells from the epiblast. So the epiblast is going to play a really important role. We're going to see that later. Okay. And the hypoblast, the one that we have here is the hypoblast, and it's going that way, does not contribute any cell to the development of the embryo at all. Instead, the hypoblast form the external membranes that we saw prior, which include portion of the yolk sac, the stalk that links the yolk mass to the endodermal digestive system. So this is the gastrula, which implicates the hypoblast. This left column here shows the mid-sagittal section through part of the blastoderm. This middle column here shows the entire embryo view from the ventral side, displaying the migration of the primary hypoblast and secondary hypoblast, okay, which will become the endoderm cells. And the right side column shows the entire embryo from the dorsal side, okay? This is the mid-sagittal. So in A, we see the delamination of the cells from the zona pellucida to form the hypoblast. We also see the aggregation or congregation of these cells. And we see also the appearance of the coracycle cells that start aggregating and becoming more prominent, okay? Here we see These are the hypoblast population island. This is the color cycle forming. And this is the dorsal. In B, that's the stage 12, we see the formation of the primary hypoblasts. So become more congregated in that subgerminal cavity in here. Okay. And normally it's more on the anterior extension of the color cycle. So we see how the color cycle continues, start going up, and it's picking up all these hypoblast islands, right? It's picking them up as it goes. In C is the formation of the secondary hypoblast with a full connection. So now we have gone all the way up, and we see how this section of the color cycle becomes more prominent into that little part of the embryo. Now we come to form a structure that is very important for avians and mammals, and is the primitive streak. Okay, so the major structural characteristic of reptile, birds, and mammal gastrulation is the formation of this primitive streak. Okay, so here in D, we see the appearance of the primitive streak cells. Okay, that one in red, to form the third layer that is placed between the epiblast and the hypoblast. So here we have, this is the epiblast, this is the secondary hypoblast, and the cells start appearing here near where we have the cycle. Okay. So in E, in here, we see that the primitive streak becomes the definitive region of the epiblast, okay, with the cells migrating through, become the mesoderm and the endoderm. So here again, we see the thickening appearing in the posterior part of this primitive streak cells, and then they go and start projecting towards the front of the embryo. And that now going to become the mesoderm and the endoderm. And I just seen that is 11:18. So we're going to stop there because the next slides are to be looking into the gastrulation and how this primitive streak allow some cells to gain the insight to form the rest of the embryo. So with that, where is my mouse? There we go. I don't know, I can't do anything. Okay, there we go. Okay, come on, camera. Here we go. We're gonna continue on Tuesday, this lecture, and I'm gonna see you tomorrow. I saw that a lot of you have read part of some of the papers. There are still a lot of people that haven't participated into their groups for the article discussion in the perusal section. Remember that 6% of your mark is this thin. I try to make it simple. I'm trying to put papers that are of interest. I have seen some of the comments on the papers like, oh my God, I didn't know. Somebody say, I have my laptop right now on my lap. So these papers are always meant to incite your interest and your curiosity, and that is relevant to all of us. And the reason why I only put male stuff is because when we talk about infertility, 99% of the case, who is the culprit or signal as the culprit? The females. Or now we know males also have a lot to do with it. Okay, so this is my feminist part, so please, males, don't take it wrong. It's just to open the eyes to this point that normally when we talk about infertility, most of the cases we are looking at what is wrong with her, right? And only when they say, no, she's okay, it's like, oh, I may be the wrong, so then is when they need to provide some sperm sample. to see what is wrong with him. But it's more than that, right? It's our lifestyle, it's a lot of the environmental circumstances that we're exposed to. So the reason why I put those two papers, they are very relevant because we live in a very toxic environment. We don't live in a bubble. We live in a very toxic environment. So having said that, if you haven't read your papers, please go and read them. They are very short this time, okay? And comment. The more you comment, the more you interact with one another, the better chances you have to get your points. Okay, if you don't participate, if you don't read them, that will be a 0 on a 6%. It's an easy way to win it. Okay, so see you tomorrow then, guys. Have a nice end of afternoon today.